cell organelle is endoplasmic reticulum. So this is its structure. So yeah, we all know that rough endoplasmic reticulum, it is directly attached to the nuclear envelope. Okay, so these are the components of the rough endoplasmic reticulum with the ribosomes. Yeah, this is another diagram. So it is, of course, every cell organelle is an important cell organelle. It is a network of membrane-bound cavities, vesicles, and tubules. Okay, so it was identified by Garnier. Okay, and the name endoplasmic reticulum was given by Porter, Keith Porter. It extends from nucleus to almost cell membrane. Cell membrane వరకు ఉంటుంది దాదాపుగా. So D. Robertis, Nowinski, and Sage. So all of them, they called it as cytoplasmic vacuolar system. ఎందుకంటే చాలా మటుకు vacuoles అనేటువంటివి వీటిని సరౌండ్ చేస్తూ ఉంటాయి. So it is absent in egg cells and embryonic cells and mostly matured mammalian RBC. It is a simple sac of hemoglobin and other some proteins and enzymes, but it doesn't contain any cell organelle. And of course, all the prokaryotic cells, they lack the major cell organelles except 70s kind of ribosomes. It is a single cell membrane or single membrane bound cell organelle. Okay, so manaki mitochondria, chloroplast చూస్తే అవి double membranous covered structures, but here it is single membrane bound cell organelle. So yeah, it is, its number is more in actively protein synthesizing cells. సో ఏ సెల్స్లోనైతే ప్రోటీన్ సింథసిస్ ఎక్కువగా ఉంటుందో వాటిలో వీటి సంఖ్య అనేది ఎక్కువగా ఉంటుంది. సో ఇట్ కన్సిస్ట్ ఆఫ్ 3 కాంపోనెంట్స్. వన్ ఈజ్ సిస్టర్నే, వెసికిల్స్ అండ్ ట్యూబ్యూల్స్. యా, సిస్టర్నే, దీస్ ఆర్ ఆల్ ది సిస్టర్నేస్. ఓకే, సో లాంగ్ ఫ్లాట్ ఎండ్ అన్బ్రాంచ్డ్ సాక్ లైక్ స్ట్రక్చర్స్ అరేంజ్డ్ యాజ్ ప్యారలల్ బండిల్స్, ఒకదాని తర్వాత ఒకటి ఒకదాని తర్వాత ఒకటి. And they may contain ribosomes here sometimes. Okay, that means in rough endoplasmic reticulum, ribosomes are present. In smooth, they do not contain ribosomes. So they are also found more in secretory kind of cells. So vesicles, here the vesicles are not given here. So actually vesicles are present like this surrounding the, you know, these cisternae. Okay, so which are the products of these cisternae? So they are round or ovoid structures. They are abundant in glands like pancreas, okay, or pancreatic cells. Yeah, they are found at the end of the cisternae and tubules. ఎందుకంటే ఈ సిస్టర్నే అండ్ ట్యూబ్యూల్స్ నుంచే వెసికల్స్ అనేటువంటి ప్రొడ్యూస్ అవుతాయి కాబట్టి. సో హియర్ దీస్ ఆర్ ది ట్యూబ్యూల్స్, చిన్న నాళాల లాంటివి, స్మూత్ వాల్డ్ అండ్ హైలీ బ్రాంచ్డ్ స్ట్రక్చర్స్. సిస్టర్నే ఆర్ అన్బ్రాంచ్డ్, ట్యూబ్యూల్స్ ఆర్ బ్రాంచ్డ్. సో ట్యూబులర్ స్పేసెస్ విత్ ఇన్ ట్యూబ్ కాబట్టి లోపల స్పేస్ ఉండి హ్యావింగ్ డైవర్స్ ఫార్మ్స్, డిఫరెంట్ షేప్లో ఉంటాయి. Present in non-secretory cells like muscles. Okay, which store, the calcium, we all know that. So endoplasmic reticulum are 2 types. So rough and smooth endoplasmic reticulum. So rough endoplasmic reticulum, because it contains ribosomes, whereas smooth without any ribosomes. So they are present in fat cells, steroid hormones synthesizing cells. Smooth endoplasmic reticulums are present in fat cells and steroid hormone synthesizing cells. In retinal cells, they are called as myeloid bodies. In muscle cells, they are called sarcoplasmic reticulum. Yeah, what does ER consist of? Endoplasmic reticulumలో మేజర్గా ఏముంటాయి? They contain phospholipids, inositol, lecithin, sapalins, enzymes like steerases, NADH, cytochrome C reductase, NADH diphorase, and glucose 6-phosphatase, and of course ATPases are also present. So there are two kinds of ATP synthetases or ATP enzymes. One is ATPases. ATPase means so they they can synthesize ATP. Whereas ATP synthetases, they are exclusively participate in ATP synthesis. Okay, so then uh, yeah, small particle fraction obtained by high speed centrifugation of cell homogenate. So uh, these are also microsomes are small vesicles. They are part of the endoplasmic reticulum. So they are formed in a very small fraction. So when we centrifuge the endoplasmic reticulum with high speed centrifugation from a cell homogenate, so it is found by the clot. So what is the specialty of microsome? They contain peroxidase, which are very, very important for the intracellular digestion, intracellular killing of microbes. Okay, so but later these peroxisomes and glycosomes. Okay, so they were exclusively present in the plant cells only. Particularly these glycosomes, they are exclusively present plant cells, and later it was confirmed that they are not part of the endoplasmic reticulum. Yeah, peroxisomes, they are present in most of the protozoan animals. Okay, and also in some human cells. And then annulate lamellae, membranous flattened sacs containing annuli and pores. ఇవి కూడా కొంచెం అటు ఇటుగా సో ఇలాంటి సిస్టర్ని లైక్ స్ట్రక్చర్స్. సో దే ఆర్ కాల్డ్ annulate lamellae. They are endoplasmic reticulum with annuli, కొంచెం రౌండ్ షేప్ లో, అండ్ పోర్స్ లైక్ న్యూక్లియర్ మెంబ్రెయిన్. ఓకే, అంటే కొద్దిగా అంటే దిస్ ఈస్ annulate lamellae. దే దే సజెస్ట్ దట్ ద ఎండోప్లాజమిక్ రెటిక్యులం ఆరిజినేటెడ్ ఫ్రమ్ ది న్యూక్లియర్ ఎన్విలోప్ అనే దానికి annulate lamellae ఒక ఎగ్జాంపుల్ అకార్డింగ్ టు మెక్కల్లక్. So anyway, oocytes, spermatocytes, embryonic cells, tumor cells, and invertebrates, they contain these annulate lamellae. They are the intermediate stages of endoplasmic reticulum. So they are originated from the nuclear envelope. So anyway, origin of endoplasmic reticulum, there are many theories are proposed about the origin of endoplasmic reticulum. Some say they are formed directly from the protoplasm. So Palade, according to Palade, so it was produced from the plasma membrane, but most people believe that endoplasmic reticulum, okay, its biochemical structure is almost similar to nuclear envelope. So most people believe that endoplasmic reticulum is derived from the nuclear envelope. So functions of endoplasmic reticulum are very, 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 very important. First thing, just like cytoskeleton, endoplasmic reticulum, it gives mechanical support to the cell. It helps for the intracellular transports. They are called as protein factories. That means because the ribosomes present on the endoplasmic reticulum, so that's why the protein synthesis occurs on the membranes of the endoplasmic reticulum. That's why they are also known as protein factories, and they produce micro bodies with peroxidase enzymes, which are needed for the killing of the microbes. And the smooth endoplasmic reticulum is an important site of cholesterol and steroid hormones. This is very, very important point. Smooth endoplasmic reticulum is the site of cholesterol and steroid hormone synthesis. And in rough endoplasmic reticulum, there is a process called glycosylation takes place. What is glycosylation? It is nothing but the addition of carbohydrates to protein. This is a kind of editing after protein formation. Protein తయారైన తర్వాత అవి they are fallen into the cavity of the or lumen of the endoplasmic reticulum, where the proteins are the some additions are occurred in the lumen of the endoplasmic reticulum. Among such is glycosylation. Glycosylation అంటే ఏం లేదు, ప్రోటీన్కి కొంత కార్బోహైడ్రేట్ పార్ట్ యాడ్ చేయడమే glycosylation. There is an enzyme called glycosyl transferase enzyme catalyzes this reaction. And one more famous, very famous function of the endoplasmic reticulum is detoxification. 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 What do you mean by detoxification? So detoxification is the process by which the toxic substances are converted into less toxic or okay, sorry, there was network problem. Anyway, yeah, it gives mechanical support. Endoplasmic reticulum gives mechanical support to the cell. It acts as an intracellular transport system or circulatory system. Protein synthesis occurs on rough endoplasmic reticulum. They also produce microbe bodies which contain the peroxidase enzymes. And then this is very important, synthesis of cholesterol and steroid hormones occur in endoplasmic reticulum, smooth endoplasmic reticulum. This is very, very important point. Glycosylation. What is glycosylation? Addition of carbohydrates to protein that occurs within the cavity of the rough endoplasmic reticulum. So glycosyl transferase is an enzyme that catalyzes this reaction. What is detoxification? Detoxification is the conversion of toxic substances into less toxic or no toxic substances. Okay, so this particular thing occurs especially in liver cells. Liver is the place where all the toxic substances are converted into non-toxic substances or less toxic substances. So that process is called metabolism of xenobiotics or the detoxification. Even alcohol is also detoxified, changed to non-alcoholic or less toxic substance in the liver. In the liver cell, the harmful chemicals are neutralized in smooth endoplasmic reticulum. And of course, lipid synthesis, triglyceride synthesis also occurs in the smooth endoplasmic reticulum. And glycogenolysis. మనకు గ్లైకోజెనాలిసిస్ అనేది, గ్లైకోజన్ అనేది డిగ్రేడ్ అయ్యి గ్లూకోజ్ అనేది ఫామ్ కావడం. సో అది జనరల్గా మోస్ట్లీ ఇట్ అక్కర్స్ ఐదర్ ఇన్ లివర్ ఆర్ ఇన్ మజిల్. ఓకే, సో in muscle cells and in liver cells, endoplasmic reticulum is the site of the glycogenolysis by the help of enzyme glucose 6 phosphatase. And in muscles, it is the place of storage of calcium ions. So these are the functions of what we call the endoplasmic reticulum. And there are some other points like protein folding, quality control. Okay, newly synthesized polypeptide chains are folded into their functional three-dimensional shapes with the help of chaperone proteins. Just wait. Okay, the PDF. Okay, so here, yeah, so smooth endoplasmic reticulum, it 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 is the site where the uh, folding of proteins with the help of molding proteins called chaperones. చాపరాన్స్ అని పిలువబడేటువంటి ప్రోటీన్స్ సహాయంతో అప్పుడే ఏర్పడిన న్యూలీ ఫార్మ్డ్ ప్రోటీన్స్ అనేటువంటి వాటి ఒరిజినల్ త్రీ డైమెన్షనల్ స్ట్రక్చర్లోకి అవి మౌల్డ్ చేయబడతాయి, ఫోల్డ్ చేయబడతాయి. And it also plays an important role in carbohydrate metabolism. So these are the functions of the, endoplasmic reticulum. And then Golgi complex. So it is otherwise known as Golgi body or Golgi apparatus. It is more or less similar to the, what we call the endoplasmic reticulum. Okay, so it also a cell organelle which mostly produces the transport vesicles or secretory vesicles. Anyway, so Golgi complex, George Camillo Golgi in 1893 discovered in Purkinje cells of oval and cat brain. So Purkinje cells అనేటువంటి special cells, they are present in the brain. And then it is a two membranes bound cell organelle. Okay, we don't, in general, we don't, we cannot identify two membranes while watching the, Golgi complex, but it has two membranes. Then Camillo Golgi identified it by using silver nitrate. So other names of Golgi are tropospongium, older names. Okay, according to a scientist called Pearl, lipochondria and Baker's bodies. So in some plants, they are called dictyozoles. As usual, they are absent in prokaryotic cells and in matured mammalian RBCs, and where they are present in abundantly. So they are present in abundantly in secretory cells like gland cells, okay, and food storage cells. So they are present in high number in secretory cells, which secrete different kinds of hormones, enzymes, etc. In those cells, the Golgi complex are more in number. They are also present in the cells in which the food is stored. Yeah, as usual, just like, rough endoplasmic reticulum, these also do contain cisternae, vesicles, tubules, and Golgi vacuoles. Yeah, so Golgi is surrounded by two plasma membranes. Okay. సిస్టర్నే. సో ఇప్పుడు మీకు కనబడేది ఏదైతే ఉందో, సో దీస్ ఆర్ స్టాక్, అంటే దొంతర అని అంటాం. ఒకదానిపైన ఒకటి అమరి ఉన్నటువంటి ఫ్లాట్ ప్యారలల్ స్టాక్ ఆఫ్ మెంబ్రెన్స్. దే అరేంజ్డ్ వన్ అబౌవ్ ది అదర్. Intercisternal material that bind them. All the cisternae are connected by intercisternal material. They are slightly bent. మనం అబ్జర్వ్ చేస్తే యాక్చువల్గా గాల్జీ అనేది కొంచెం కొద్దిగా బెండ్ అయి ఉంటుంది ఇట్లా. ఓకే, సో దే ఆర్ స్లైట్లీ బెండ్. సో దే హ్యావ్ ఏ కాన్వెక్స్ అండ్ కాంకేవ్ ఏరియాస్. సో దిస్ ఈస్ కాంకేవ్ ఏరియా, పుటాకారం ఉంటుంది. సో దిస్ ఈస్ కాన్వెక్స్ ఏరియా, సో కుంభాకారం అని అంటాం. సో జీసీ ఈజ్ ద ఓన్లీ సెల్ ఆర్గనల్ దట్ షోస్ పోలారిటీ. ఇది ఒకటి గుర్తుంచుకోండి. సో పోలారిటీని ప్రదర్శించే వన్ అండ్ ఓన్లీ సెల్ ఆర్గనల్ ఈజ్ the Golgi complex. And then అంటే అన్ని కణాంగాల్లోకెల్లా అన్ని సెల్ ఆర్గనల్స్లోకెల్లా సో 2 డిఫరెంట్ షేప్స్ ఉన్నటువంటి డిఫరెంట్ అంటే పోలారిటీ అంటే ఏంటంటే డిఫరెంట్ ఎండింగ్స్ ఉండడం. సో పొలారిటీని చూపే ఏకైక సెల్ ఆర్గనల్ ఏది అంటే అది యునో వాట్ వి కాల్ ది Golgi apparatus. The convex part is called cis Golgi or forming phase. సో ఇది ఏదైతే ఉందో ఈ కుంభాకారం ఏరియా దీన్ని సిస్ గాల్జీ అని పిలుస్తాం. ఇక్కడ నుంచి సీక్రెటరీ వెసికిల్స్ అనేటువంటి వస్తూ ఉంటాయి. ఇట్ ఈస్ టువర్డ్స్ ద న్యూక్లియస్ అండ్ న్యూ సిస్టర్నే ఆర్ యాడెడ్ ఫ్రమ్ ఈఆర్. ఎండోప్లాజ్మిక్ రెటిక్యులం నుంచే ఇది ఏర్పడుతుంది. కొత్త సిస్టర్నే అనేటువంటివి ఈ ఫార్మింగ్ ఫేజ్లోనే యాడ్ అవుతాయి. ద కాంకేవ్ పార్ట్ పుటాకార తలం ఏదైతే ఉందో సో దట్ ఈస్ కాల్డ్ ట్రాన్స్ గాల్జీ. So here large secretory vesicles are formed. So small then after cistern vesicles, small droplet-like structures about 40 angstrom in diameter, present at periphery of the Golgi complex. So by budding, they are formed from the cistern. Cistern అనేటువంటివి pinched off or budding లాగా అవడం వల్ల సో ఈ యొక్క వెసికిల్స్ అనేటువంటివి tubules from the trans phase of Golgi complex has the many small GC has many small tubules arise and interconnect cistern vesicles and vacuoles. So trans phase నుంచి అంటే పుటాకార తలం వైపు నుంచి సో జీసీ హ్యాస్ మెనీ స్మాల్ ట్యూబ్యూల్స్ లాంటివి ఉంటాయి. అవి దే ఇంటర్కనెక్ట్ సిస్టర్నే vesicles and vacuoles. These transport material is among them. There is a continuous disturbance in the network. The tubules, they are formed from the concave area or పుటాకార తలం నుంచి ఏర్పడితే చాలా ట్యూబ్యూల్స్ ఉంటాయి. సో అవి ట్యూబ్యూల్స్ అనేటువంటి సిస్టర్నేల్ని వెసికిల్స్ని వాక్యూల్స్ కలిపి ఉంచుతాయి. అండ్ గాల్జీ వాక్యూల్స్, these are large, spacious, round sacs found at the edges of cistern. Cistern యొక్క edge దగ్గర. So they are formed. Okay. So large, spacious, round sacs found at the edges of cistern. They are formed by the expansion of cistern. So chemical composition, it is made up of about 60% protein, 40% lipids. It contains phospholipids like cephalins and lecithins on the membrane. ADPase, and thymine pyrophosphates, acid phosphatase, uridine diphosphate, N-acetyl glucosamine transferase, and according to Kane, carotenoids so are also present. He found them in the limnia, a kind of slug. How the Golgi complex is originated? So from endoplasmic reticulum, according to Pallad, Beams, and Kessel. So literally it is formed from the endoplasmic reticulum. So yeah, we have seen during the formation of spermatozoa. So there is an important structure called sperm's acrosome is formed from the Golgi complex. During spermatogenesis, proacrosomal granule is formed. From that acrosomal granule is formed. So from that acrosome is formed. So these are the steps involved in the acrosome formation. And then it also participates in the cell wall formation. You remember, particularly the formation of mid plate, మధ్య ఫలకం ఏర్పడంలో ముఖ్య పాత్ర వహించేది గాల్జీ కాంప్లెక్స్. ప్లాస్మా మెంబ్రేషన్, ఒక ఎగ్జోసైటోసిస్ జరిగిన తర్వాత కొద్ది మొత్తంలో ప్లాస్మా మెంబ్రేన్ పోతుంది. సో దాన్ని రీకూప్ చేసేది, తిరిగి కాంపెన్సేట్ చేసేది కూడా, కొత్త ప్లాస్మా మెంబ్రేన్ ఏర్పరిచేది కూడా ది గాల్జీ కాంప్లెక్స్. So carbohydrates of plasma membrane synthesized by the Golgi complex. And also the most important function of Golgi complex is formation of lysosomes. What is the difference between other simple vesicles and lysosomes? Lysosomes కూడా ఒక రకమైన వెసికిల్సే. అయితే వీటిలో యాసిడ్ హైడ్రోలైటిక్ ఎంజైమ్స్ ఉంటాయి. యాసిడ్ హైడ్రో right. So secretion. This is very, very important function of the Golgi apparatus. All the gland cells, including mucus, sweat, hormonal, enzymatic, nerve cells, okay, that secrete neurotransmitters, sebaceous glands, antibodies, mammary glands, also done by Golgi. Okay, so secretory cells are the glandular cells like mucus gland cells, sweat gland cells, enzymatic nerve cells that secrete neurotransmitter. Okay, sebaceous glands of our skin and antibodies and mammary glands also. All these kind of secretions are the so secreted by the presence of the Golgi complex only. And concentration and storage of three secondary products by losing water. Okay. Concentration and storage of secondary products. Okay. కొన్ని ద్వితీయ మెటబాలిక్స్ అనే కొన్ని ఉంటాయి. అవి ఇందులో నిల్వ చేయబడతాయి. సో దెన్ గ్లైకోజిలేషన్ విచ్ ఈస్ అడిషన్ ఆఫ్ కార్బోహైడ్రేట్స్ టు ప్రోటీన్ ఆర్ బిల్డ్ lipid. So in just like rough endoplasmic reticulum, so in Golgi complex also glycosylation occurs. Okay, so then sulfation. The goblet cells of the intestine produce mucigen as a mucopolysaccharides. The goblet cells of intestine contain mucigen అనేటువంటి ఒక మ్యూకోపాలిసాకరైడ్, అంటే ఒక రకమైన గ్లైకోల్ ప్రోటీన్ అన్నమాట. So the GC adds sulfate to glycoprotein to form mucigen. The goblet cells of intestine produce mucigen, a mucopolysaccharide. Okay, so which is produced from the Golgi complex. The GC adds sulfate to glycoprotein to form mucigen. Okay, then sulfate అనేది యాడ్ చేయడం వల్ల that will become a glycoprotein. Okay, so then the enzyme was sulfate transferase, lipid attacking and secretion. So, sorry, lipid packaging, protein packaging also. Okay, lipid packaging and protein packaging also occurs by the Golgi complex. So, in intestinal cells, and conversion of pro-hormones into active hormones. Okay, hormones అనేటువంటివి inactive form లో ఉంటాయి. They are converted into active forms. Okay, so anyway, the major function of the lysosome is the secretion. Secretion is its major function. So, okay, yeah, lysosomes. So lysosomes were first discovered by Christian Didouet. They are single membrane bound cell organelles and it is a lytic body. ఇది విచ్ఛిన్నం చేసేటువంటి సెల్ ఆర్గనెల్. Its common name is suicidal bags of cells. Old name is pericanaliculair body. So they are abundant in cells which are related. Yeah, they are abundant in cells in which related with the enzymatic activity. Enzymatic activity ఎక్కువ ఉన్న cells లో ఇవి సంఖ్య ఎక్కువగా ఉంటుంది. Example, liver, pancreas, kidney, spleen cells. There's simple spherical bodies. Okay, so lysosomes are simple spherical bodies. They are nothing but the Golgi vacuoles having acid hydrolytic enzymes. They contain large number of dense granules having acid hydrolytic enzymes and digestive enzymes. They are polymorphic cell organelles based on stages of digestion. సో మనం చూసినాం ఇవన్నీ కూడా ఇక చూడండి ఇది లైసోజోమ్స్. సో దే ఆర్ యాక్టింగ్ ఆన్ సో phagosome, right? So then types of lysosomes. There are, four kinds of lysosomes. Primary lysosomes, like small sacs with acid hydrolytic enzymes. Small sacs with acid hydrolytic enzymes produced in GC. Okay, Golgi complex. Okay, and they are like storage vesicles of enzymes. And then secondary lysosomes. So here the food vacuoles or digestive vacuoles are called secondary lysosomes. మనం ఆల్రెడీ ఇందాక చూసిన ఫాగోసైటాసిస్లో ఎప్పుడైతే దే స్టార్ట్ consuming the food material. Okay, so the food vacuoles are digestive vacuoles are formed, okay, within the cell. Okay, so secondary lysosomes are nothing but the food vacuoles in which the food is being digested. You can also call them as digestive vacuoles. And then when an endosome or phagosome fused with primary lysosome, that is called secondary lysosome. Okay, so మన బాడీలోకి ఫాగోసైటాసిస్ లో భాగంగా ఏదైనా ఫారిన్ బాడీ రాగానే ఫస్ట్ దాని చుట్టూ సో yeah, so there are four kinds of lysosomes. Primary lysosomes are actual lysosomes having acid hydrolytic enzymes. So they are like storage vesicles of enzymes. Secondary lysosomes are nothing but the food vacuoles which are mixed with the phagosome and with a lysosome. That is called secondary lysosomes. They are digestive vacuoles. They also participated, phagocytosis. Cell. Okay, so during phagocytosis, the entered foreign body is digested, killed by the lysosomes with its acid hydrolytic enzymes. When an endosome or phagosome fuses with primary lysosome, that is called secondary lysosomes. So residual bodies. So secondary lysosomes with undigested waste ని మనము ఏమని పిలుస్తాము? రెసిడ్యువల్ బాడీస్ అంటే ఆటోఫాజిక్ వెసికల్స్. Lysosomes with self cell parts being digested inside. Autophagic vacuoles అంటే ఎప్పుడన్నా సెనసెన్స్ ఆర్ ప్రోగ్రామ్డ్ సెల్ డెత్ వల్ల సెల్ డై అవ్వడం. సో దట్ ఈస్ ఆల్సో డన్ బై ది these lysosomes which are aptly called as autophagic vacuoles. Yeah, its stability of lysosomes is dependent on the hormones inside it. Cortisons, corticosterone, cholesterol, glucagon, vitamin A, E, pancreatic salts, progesterone. So all these, okay, they so anyway, pancreatic salts, progesterone hormone, all are present inside these lysosomes. Yeah, chemical composition, about 50 enzymes are isolated. Most of them are acid hydrolysis enzymes, no oxidation enzymes. So example, acid ribonuclease or DNase, acid phosphatase, then esterase, which breaks the ester bond, cathepsin, which breaks the protein, chain. Okay, and collagenase, having high amount of collagen. Then phospholipase. So phospholipase is also one kind of secretory or cathepsin, collagenase, phospholipase, peptidase, sulfatase. ఓకే, ఇవన్నీ కూడా రకరకాల ఎంజైమ్స్, అవన్నీ కూడా డైజెస్టివ్ ఎంజైమ్స్గా మనం కన్సిడర్ చేయొచ్చు. ఆర్జిన్ ఆఫ్ lysosomes. So lysosomes are directly originated from the Golgi complex. Okay, so but according to Novikov, they may also form from ER. సో ఇవి కూడా ఈఆర్ నుంచి ఏర్పడతాయని కొంతమంది శాస్త్రవేత్తల భావన. ఫంక్షన్స్. సో ఫాగోసైటాసిస్. ఓకే. సో మన న్యూట్రోఫిల్స్, మాక్రోఫేజ్ అనేటువంటివి భక్షణ చర్యలో లైసోజోమ్స్నే వాడుకుంటాయి. సో ఆటోఫాగి. సో హియర్ హియర్ ఇట్ ఈస్ ద eating of own food material, own body parts. Okay, so that is called autolysis. So in metamorphosis, the autolysis is an important process in the many larval adults. Among them, sorry, autolysis. Whenever the cell gets the signal to stop working, so it will break all the lysosomes, and it breaks. Okay, so then extracellular digestion. It also participates by releasing enzymes externally also. So fertilization. Golgi complex produces the acrosome in sperm is a modified lysosomes. Okay, so Golgi complex produces the acrosome, which is a modified lysosome. So harmful effects of lysosomes. So lysosomes are damaged when silica accumulation in lungs. Such lysosomes damage the lung cells. DNAs of lysosomes, if leaked, can cause deletion mutation. Sometimes it leads to leukemia. So lysosome lo ఉన్న DNAs అనే enzyme. So if at all it is leaked, it causes deletion of maturation. Sometimes it may lead to leukemia. Sometimes lysosomes do not fuse with phagosomes. If those phagosomes contain TB or leprosy bacteria, that leads to TB or leprosy. Okay. So then microbodies are peroxisomes. So here, okay. So yeah, microbodies, they're also known as peroxisomes. So they're also known as uricase granules, amino oxidases, catalases, and all those. So these are the